Okay. Yeah, Monday. Where did you go? I didn't put that there. I don't know what that is. I mean, I know what it is. It's from, obviously it says on it all.
Yes.
So I don't know how that got saved on there. So I'm gonna need to remove it, but
Yes. Okay.
absolutely 100%. You need to be going off of the learning objectives.
Okay.
The ones that are like compare and contrast or explain or
Yes.
whatever. These are kind of hard to do, aren't they? What is it? Okay, no, these are the Blues challenge things.
Yes.
Right, so this learning objective, this is like literally where the test questions are gonna come from. I use this to like ask questions to test your knowledge.
Okay,
Like
so
study
this
tools.
is like just what
These
I
are
should.
absolutely what you need to know, and this
Okay.
is just like checking a different way of asking.
Like a check it off.
Yeah.
Gotcha. Okay.
Thank you. Okay, yeah. My title did not change that, yeah. Yeah. Cool. You are gonna send this one? Okay. Yes, and I am gonna record it and I am gonna post it. Okay, hopefully we'll have at least a couple people hanging out so that like. But if there's days where nobody shows up, I'm still gonna do one. I mean, like, it's been working out for me though. There we go. So on my Canvas, on slides for this, you go to discussions, and then we have, I don't want to see that. I don't want to see my face up there. Okay, there we go. Okay. Like, I don't want to see my face up here. I know I'm muted. I want to stay muted. Okay. It's recording it from here. So, okay. So weekly review, study aids, not required in the discussion section. And then there should be in the reply for every week, I'll have the slides. And then eventually when we're done, I'll post the recording, and usually whatever AI summary Zoom gives me, I'll post it. I don't edit it. So it might have like weird things about me talking about getting my nails done or something like that if I go off track, but FYI. So I'm gonna download from here. Okay, save to files. Week 1, so I can find it in lecture. All right, now I'm going to PowerPoint because I have an iPad and it doesn't open it automatically. Files. Cloud work lecture week 1. Okay. Right. Let's do it. Okay. So we're going to do, this doesn't have chapter 4 in it. There it does. Okay. It just wasn't on the front page. Okay. So we're going to cover the key concepts for chapters 1 and 3, which are kind of together, right? The lecture is a together lecture and the slides are together. Then we'll do chapter 2, which is our chemistry review, and then chapter 4, which was our bacteria. And it's okay if you haven't read those chapters yet, but this will at least get us thinking about some of the concepts that people are missing. So for chapter 1, some of the key concepts that we covered, we talked about the definition of microorganisms. Basically, microorganisms are any organisms too small to be seen with the naked eye. It really is that simple. Microbiology is the study of microorganisms, anything too small to be seen with the naked eye. Acellular microbes, the ones that we talked about in chapter 1, these will be our viruses and our prions. In chapter 7, we'll talk about what viruses are and basically what the parts of the viruses are and a little bit more information about viruses, as well as prions. We'll talk about them too, but real basic stuff here. A virus is essentially something that has a nucleic acid, but it doesn't have all the qualifying aspects of what we consider to be a cell. So it doesn't have necessarily a membrane around it. Some of them do have membranes, so that's where it gets a little bit tricky. But they can't self-replicate, and cells can self-replicate. They can always make a copy of themselves. Now, if I take a cell off of your skin, can I make a whole new organism from that? Not without some science, some real science forcing it to do that. But can the cells on your skin self-replicate themselves? Yes. So that's what we're looking at there. So that's what makes a cell. If the cell can self-replicate itself through mitosis or binary fission, then that's a cell. Viruses can't do that. They can't do that on their own. They have to get inside of your cell and use the machinery of your cell to make copies of themselves. So that's kind of why viruses are acellular. They don't have the qualifications of what makes a cell. A prion is literally just a misfolded protein. It is. That's what it is. So it's a protein that has a normal function in your body. It does something in your body, but it got misfolded in a way that makes it prefer that misfolded kind of shape. And then it causes your other normal proteins to misfold when they come into contact with it. We see a similar effect of this. It's not quite the same, but we see a similar effect of this in Alzheimer's disease, where proteins in the brain will kind of misfold a little bit due to age. And that's going to cause them to aggregate in the brain and cause brain damage. This happens with prions, but it's not from age. It's just from being introduced a misfolded protein into your body. We'll talk about some examples of this later on. It's not common. They're very rare, but we do see them in nature. For example, with mad cow disease, that's what that is. It's a misfolded protein in cow nervous tissue that causes it to misfold in the cow nervous tissue, but it's also similar to a human protein and can do the same thing to humans as well, which we call variant Creutzfeldt-Jakob disease. And then those proteins will aggregate in the nervous tissue and cause spongiform encephalopathies. We'll learn about this in detail later on, but that's what they are in case you're wondering. And I feel like that's fascinating, but depending on which version of the video or which version of the textbook or which version of science you're talking about, eukaryotes or prokaryotes. Come on, my hand is touching my screen and it doesn't want to write. Why is it doing that? Well, whatever, I'll just scratch it out. Prokaryote. They changed the word like a year ago. So I don't care if you know it as prokaryote or if you know it as eukaryote, but on my test, I might say eukaryote. So just know that that's the same thing as prokaryote. A means without or pro means before. So these are organisms that these are the first organisms probably on the planet that we can say are cellular organisms. These have no true nucleus, but not just no nucleus. They have no membrane-bound organelles of any type. If you haven't done chapter 5 yet, that might be a vague idea for you, but chapter 5 will kind of clarify what the organelles are. So this includes an organelle would be things like your nucleus, your endoplasmic reticulum, your mitochondria. Those are all membrane-bound structures that have functions in cells that eukaryotes have, but not necessarily, but the eukaryotes or prokaryotes never have. And it's like the defining characteristic of them. It talks about it more in chapter 5. And again, these are like the simplest versions of what a cell can be. So we believe that these are likely the first organisms on the planet. They're major contributors to the atmosphere on Earth. And so back in the day, we used to have no oxygen in our environment whatsoever. All evidence of studying the Earth and the deepest cores of sedimentation and rock and everything like that shows that there was no oxygen ever on this planet until a certain time in the history of our Earth. And then what happened was microbes learned to use substances on Earth and converted it into oxygen. And literally all the oxygen on the planet came from microbes, not because it was already here, but because it came from the microbes metabolizing other materials on Earth and turning it into actual atmospheric oxygen. And now that's maintained not just by microbes, right? Now that's maintained by microbes as well as like plants. But still a good percentage, it's somewhere around 70 to 80% of all oxygen made on our planet is still made by what we consider microorganisms, including algae and planktons and things like that primarily. We don't think of them always as being there, but they're there, providing most of the oxygen on the planet. So as much as we love the rainforest to talk about how it provides a lot of our oxygen, it's actually thanks to microbes. Not saying we shouldn't save the rainforest, but you know. All right, pathogens. Pathogens are microbes that are disease causing. That's it. There are plenty of microbes in the world that you're never going to be encountering or that will never ever cause disease, even in any circumstance whatsoever, especially for otherwise healthy people. We're going to talk a little bit in chapter 17 about when your immune system goes wrong with hypersensitivities and hyposensitivities and what that really means. And essentially what that's talking about is like with people who don't have, they have like, I want to say, genetic defects in their immune system, and they might not have any immune system at all. So like SCID is one of the examples of that. So it's severe combined immunodeficiency. They don't have any immune system. And so they can't even like have an immune response to protect them from just bacteria in the environment. So they get sick from everything, and they usually die at a very early age. The worst version of this is X-linked SCID, which is linked on the X chromosome. Luckily, women have 2 X chromosomes, right? And so you usually have another version of that gene to make up for it. But men often only have 1 X, and they only have one, genetic men only have 1 X chromosome and 1 Y. And so if they have a problem with the gene that leads to SCID, they don't have another good version to make up for it in their genetics. And so X-linked SCID always affects men and never women. And it's the worst version you can have. And that's where boy in the bubble disease, it's a real thing. Literally can't get exposed to any bacteria, even in food, just nothing. They can't have any immune response to anything ever. And so they die very young ages typically. So you don't usually get them living up to be old enough to be like Jake Gyllenhaal in the Boy in the Bubble movie, going around and finding romance. Of course, you find out in that movie, if you've watched it, that he never had that disease. But yeah, so just interesting to think about. But even just trying to keep them completely sterile environments, I mean, it's impossible. You know, so eventually at some point they usually get sick and die of just normal stuff that you would never ever get sick from, even in the wildest imaginations. But we'll learn about that more in chapter 17. But pathogen is any disease causing microorganism. This also relates then into our disease causing microorganisms and how we thought they used to exist. It used to be believed that, you know, if you left a steak out on a countertop, and then a couple of days later, there's like maggots growing on it, that they just appeared because that's what happens over time to things. That was spontaneous generation. Because people didn't understand there were smaller things than what you could see with your eye. And now we know that what's actually happening is that flies might lay eggs on it that you can't see. And then those eggs turn into maggots and so on and so forth. Even at a more microscopic level, the whole reason that the meat would start stinking and getting slimy is because bacteria on there from the environment or from handling the meat or whatever got introduced into the meat and then just grew on it, and like a petri dish would. So microbes don't come out of nowhere. And the guy that proved that was Louis Pasteur. And he had these flasks, and like one of them was like an open flask, it looked like this. And then one of them was like a flask that had like a weird snaking neck. And like, I don't know, here's where the liquid is. This is a really terrible drawing of it. But like, this is the neck of it. And it was still open to where air could get in, but it was much, much more difficult for microbes to be able to get accidentally introduced into it. And so when he boiled each of these and to try to sterilize the liquid and then left them out on the countertop or grew them at a warm temperature. Obviously, the one that was open to the air grew stuff, but that this one never grew anything. He's like, see, it's not coming from nothing. It's coming from the air. We just can't see it. It's something. And that led to germ theory. There are germs, microorganisms, things we don't see that are already in the air that is leading to this stuff growing in media or on our foods, or that could be making us sick. And that was Pasteur's experiment. Then Robert Koch came up with these steps to try to prove where the diseases were coming from. That was Koch's postulates, which we'll talk about again later as well. But he basically showed that like if you have an organism that's sick and you can isolate the microbe from that organism, and if you can isolate it and put it into a healthy organism and it makes the same disease, you found the cause of that disease. And that's how he found the cause of anthrax and other diseases as well. But he's very well known for creating that idea of, seems so obvious and basic to us now, like these concepts, but back then they literally like thought like a bad smell made you sick, miasma and stuff like that. And that's why plague doctors wore the masks that they wore. They had the long nose on them because they stuff them with herbs, because they thought, oh, if I smell the nice thing and I don't smell the bad thing, I can't get sick. Not how it works. So not how it works. But that this is where germ theory came about, next to Louis Pasteur. And then that lead to, okay, well, if germs are causing things, how do we keep them from causing contamination into our patients who are getting sick, or into our environment when we're studying them, and et cetera, like we learned about with aseptic technique. And this was introduced by Lister into the operating rooms, where he decided to start using carbolic acid, which is phenol, to start cleaning operating rooms before he would have surgery and after he would have surgery, to sanitize that. And then we have Semmelweis and Holmes to thank for like hand washing and stuff like that. They started noticing that women who gave birth thanks to midwives were surviving birth. Women who gave birth thanks to doctors were dying. And so he looked into that, Semmelweis did, and he noticed that the doctors were performing autopsies or dealing with surgeries and things like that and never washing their hands in between. And they might like remove debris, but not washing their hands. And so he introduced the concept of hand washing to remove the germs. And lo and behold, now people are able to survive surgeries and giving birth and things like that. It's just wild to think about that was ever a thing, but it was. And so that's where like these ideas of aseptic technique, or even in practicing this in lab or in hospital settings, came to be, thanks to these people thinking about that. And when we learn in the epidemiology chapter 14 in unit 2, Florence Nightingale was a big part of that as well. So she's considered the mother of nursing. And in the Crimean War, she noticed that doctors weren't washing their hands. And we kept having all of these soldiers coming in with like stab wounds and things like that, but they're dying of dysentery. And she's like, what the heck? Like dysentery doesn't come from stab wounds. So why are these people getting sick with these diseases? And she noticed it's because people weren't washing their hands. It takes a woman sometimes, right? So so she's considered the mother of nursing. That's Florence Nightingale. So that's these. We talked about these. Okay. And then taxonomy. The best way that I can help you guys remember this is by creating like an acronym or something. So does King Philip come over for good spaghetti or something? So whatever. And so that's one that I feel like the King Philip one is one that I learned back in the day. King Philip comes over for good spaghetti was one, but now we have domain coming before that. That's a newer thing since like the 80s or something. And domain, you do need to know the 3 domains. So domain, there's 3. You are either in domain eukarya, or you are in bacteria or archaea. We're not going to see a whole lot of archaea in our microbiology studies in this class. Bacteria and archaea both fall into the prokaryotes or eukaryote group. But they are so distinctly genetically different, and the structures of their cells are so distinctly different. we are closely related, more closely related to archaea, or archaea is more closely related to us than they are to bacteria. Even though if you looked at them in a microscope, they look the same, they're same size, same structures. They don't have a nucleus, the archaea. They don't have any cell-like structures, but their genetics and their ribosomes are the same as ours, stuff like that. So it's just evolutionarily, the archaea are different. Archaea are also wildly weird. They're almost alien in a way. Whereas bacteria and eukaryotes, the ones we're going to talk about by far more, they'll have phospholipid bilayers like we talked about here, but the archaea don't. They have weird hydrocarbon like octane tails and stuff like that. So it's just wildly different structure of their cells and stuff. So we have eukarya, we have bacteria, we have archaea. We will talk about one archaea species in our diseases, and that is called Methanobrevibacter oralis. Well, the oralis probably hints at you where it's found in your mouth. And people who have particularly bad issues with tooth decay, gingivitis, periodontitis, if they have Methanobrevibacter oralis, that archaea bacteria, but not bacteria, that archaea organism, then they're at much higher risk for severe presentation of that tooth loss and having difficulty controlling bad breath and stuff like that. And it's methane producing archaea that can be part of your oral biome. Binomial nomenclature, let's not make too much of this, okay? Binomial nomenclature has some basic rules. First of all, binomial nomenclature tells you that every species is always two words. Binomial, to name, right? So, who cares? Whatever. It's two words. The first word, I feel like I'm playing charades or something, but the first word is always the genus. The second word is called the specific epithet. And that the whole thing equals the species name. The genus, the first letter is always capitalized. So for example, Homo, capital H. And then second word will be lowercase first letter, sapiens. You guys know this one. Escherichia coli is E. coli. Staphylococcus aureus, that's the binomial nomenclature. If you are typing, so this is another thing you need to know, typing, because I'm going to have you do this on the test. I don't know why I put an E at the end of typing, so let's pretend like that's not there. If you're typing, it needs to be italicized, and I will check that. You will have the ability to italicize it, but I'm not going to remind you to do it on the exam, right? I'm going to give you like an all caps name and say, type this out in binomial nomenclature. So you'll need to know, capitalize the first one, lowercase the second one, italicize it. It'll be spelled correctly. I won't make you correct spelling or anything like that, but yeah. So typing, it needs to be italicized. If you were handwriting, if for some reason your computer isn't working that day and I give you a paper test, I prefer to do that than send you to the testing center. Then if you're handwriting it, you'll need to underline it. If you're ever like, normally you're going to be taking my test on a computer and you get to a question like that and you're taking it on paper. And if you were to call me over and be like, so if I was typing this, I know I would italicize it. I know that that's correct for binomial nomenclature, but I can't remember what to do when I'm handwriting it. I won't mind reminding you then. But if you knew you when you were typing it, that's to me, it's like, hey, you got the idea. Okay, so don't forget about that too much. But if you were handwriting it, technically you would underline. Okay, all right. Moving on to chapter 3. Chapter 3, we have the 5 I's. The 5 I's really quickly. Inoculate, incubate, isolate, inspect, and identify. They're all just steps in trying to figure out what the microorganism is. Okay. And sometimes I always remember like one of them, and then I'll be like, okay, I know I always remember isolate. And so I'll have to like work back or work forward from that. I'm not sure, I don't have an acronym to help you with that necessarily, but then we talked about the different kinds of media. For sure, you're going to need to know differential and selective, and then enriched media. Differential, I'll put a D for differential. With differential media, this is basically a media where technically anything can grow. We're not inhibiting anything specific from growing, but there's color differences based on metabolic differences. Metabolic products are made that cause color changes in the media. The best example that one of the ones I can think of is with fermentation. When you ferment something, you often make acid as a product of it. And so that will cause a pH change. There's some kind of pH indicator in the media that causes a color change if you ferment something. So that's an example of that. Not causing anything, preventing something from growing or not, right? We're just looking at, oh, this changed the color because this was made or wasn't, all right? So selective media, it's going to have agents in it that favor or prohibit the growth of something or another thing. So usually something in it that prohibits growth of other things. So it prohibits the growth of A to favor growth of B. We see this with mannitol salts agar. Mannitol salts agar has literally salt in it, which is something that gram negatives don't survive well. That's an example of that. And so gram positives tend to grow better on mannitol salts agar. Talk about why that is in a little bit. All right. Then like we learned today with microscopes, the real image is made from the objective lens. And the virtual image is what we see in the eyepiece. Didn't talk about that much today, but you do need to know it for both lecture and for lab. It is in your lab notes, by the way. So what happens is when you have your slide on your stage and then the light source and the light's coming up out of here and your slide is up here and here's your little sample, that the light comes out of the sample, hits your objective lens on your little nose piece that rotates, goes through the microscope itself, and then comes out the eyepiece where there's another lens, which is, we already talked about in ours anyways, 10x, right? And so when the light comes out of that, it's passing through another lens. So here, when we're passing through this lens and the objective lens, this creates the first image. So that's the real one. The second image is made up here when we go through the second lens. We've gone through a whole other lens and changed it again. So that's why it's the virtual. Right? Oil immersion, basically because oil and glass are more similar to each other in density than air and glass. Basically, like when light passes through the glass and into the oil, it won't scatter as much. But if it passes from the glass into the air, it'll scatter due to the difference in the density of that. So oil immersion prevents light scatter. And there's detail in that light. Whenever we use oil immersion, we will have a clearer image. The word for that light scatter, by the way, there's a word for that. It's called refraction. Heat fixing, we learned about in lab today, so I'm not going to go back over it again. We talked about negative and positive stains today as well, so I'm also not going to go over that. There's the different differential stains. For chapter 3, really all you need to know is that these are differential stains and can tell us the difference. Chapter 4 really goes into the difference of what these stains do and how they work and what they're used for. But just briefly, gram stain tells us the difference between the bacteria cell envelopes. Is it going to be gram positive or gram negative? We'll go over that in chapter 4, I promise. Acid fast is going to tell us whether the bacteria has mycolic acid in its surface, which is a special kind of wax. again, that's a chapter 4 thing. If you haven't gotten to it, it'll make sense. We're going to review chapter 4 also. Endospore, we kind of talked about that a little bit. This is a stain that specifically looks for those hardy, dormant endospore structures. It'll make more sense when we get to lab 7, which we're not going to get to before the unit 1 exam, but endospores have a thick layer on the outside that just is protective for them. And so it's harder for stains to get into it. And so we use a special kind of stain that penetrates that. But it just looks for those hardy, dormant endospore structures. Dormant, protective. And then capsule, this is like a sugar and protein based sticky coating that is important for adhesion and creating biofilms, as well as for evading phagocytosis. And phagocytosis is when our white blood cells eat up the bad guys. And so sticky coating, it helps with adhesion, biofilms, and evading the white blood cells from eating them. Just put that for now, okay? All right, into chapter 2, the major concepts from chapter 2. We had covalent, ionic, and hydrogen bonds that we talked about. In a covalent bond, we are sharing electrons. In an ionic bond, we have a transfer or donation of electrons from one molecule to another. And a hydrogen bond is completely different from either of these. We don't move electrons at all. This is based entirely on attraction to a polar hydrogen. And that's always going to be positive. Always. Hydrogens are never going to have a negative polarity. Okay. Our organic molecules, these are molecules that are based on carbon and hydrogen. They always are also going to have hydrogen. Is carbon dioxide an organic molecule? No. Is calcium carbonate, is that an organic molecule? So, has to have hydrogen. Is bicarbonate, sodium bicarb. Bicarb has HCO3. Is that going to be organic? Technically, yes, technically. We're getting a little bit in the gray area there. I'm not going to ask you about bicarb, okay? I'm going to ask you about things like nucleic acids and proteins and things. Are those organic? Always. All right, polarity and hydrophobic and hydrophilic. We kind of talked about that a little bit today. But polarity happens whenever we have at least one of the atoms hogging the electron. So when you have water, oxygen, and we'll have a hydrogen, and we'll have a hydrogen, the oxygen is electronegative. And thankfully that negative in there kind of tells it where it's going to lean to. So it's going to want to be more negative. So it's going to want to hog electrons, which electrons we know they have a negative charge, just period. Electrons have a negative charge to them. So if it's hogging electrons, it's going to have a negative, what we call a dipole moment. Is how they draw it or whatever. I don't give a crap about that as long as you know it has a negative aspect to it, okay? And then like this hydrogen is going to have a positive polarity to it. I'm almost never going to draw it this way. I'm usually just going to put positives and negatives on it. But just to remind you guys from chemistry, like what they were talking about and stuff like that, that's the thing. That's the little weird squiggly D thing. But this is a polarity because oxygen will hog the electrons from the hydrogens. Hydrogens get this positive polarity to them, and the hydrogens get the positive polarity, and the oxygens get the negative because they spend more time. So one way I describe this to my students is like whenever I'm a sister, right? I'm an older sister, and my little sister Erica, just younger, and I was as bossy as you can imagine I would be. And so if we had a Barbie that we were supposed to share, who got the Barbie the most? I did. And so it's like that, like I hogged the Barbie, like the oxygen hogs the electrons. And so electronegative oxygen just spends more time with the Barbie. That's really all it is. And hydrogen gets it sometimes. But because hydrogen gets this positive to it, it can kind of create attractions to maybe other water molecules that also have an O that's a negative polarity. Let's keep up with this trend since we're already there. So these other hydrogens that are positive can interact with this negative on the oxygen. This attraction is a hydrogen bond. There's no electron exchange going on there. So this is just bringing that back to that, but also how important polarity is for those, okay? A lot. So when we talk about like your DNA or your RNA or your proteins or whatever, even sugars, do these things have oxygens in them? Yeah, those oxygens are always electronegative. Always. Always oxygen is. It's always like just a fact of oxygen. And so it's always hogging those electrons from hydrogen. So anytime you see a molecule that's an organic molecule, especially, but any molecule that has an oxygen in it, that oxygen's hogging electrons if they're attached to a hydrogen. Always. And so, like we think about with sugars, C6H12O6, we've got O's that are attached to H's. Those O's are always hogging the electrons from the H's. And so those positive H's can attract to other O's. And then why do you think that sugar dissolves so well in water? Because of that. Because we've got polarity. It's to allow the negative O's to interact with the positive H's of the water. And then the positive H's in the sugar to interact with the negative O's in the water. That polarity allows for that attraction to happen. And so things mix well with water because water has positive and negative polarities. And if you have positive or negative polarities, you interact well with that. There's a lot of exchange going on there with that attraction. And that's hydrogen bonds, man. And it governs literally everything in biology. The shapes of your proteins, your entire DNA, double-stranded molecule, is held together by hydrogen bonds. We're talking oxygens on one side with hydrogens on the other, just holding your entire DNA together. Wild. It's not even electron exchange. It's just attraction based entirely on this exact same theory with hydrogen bonding. So super important in biology. And it's also why when we talk about pH, which is our next thing, why when you have a shift in your pH in your body, that it has such a major effect to even have slight shifts in your pH. So if we're talking about your body's supposed to be 7.35 to 7.45, well, if you get below that 7.35 to the 7.25 range, you got too much acid in your body. What do you have too much of? Too many H pluses floating around that interacts with that hydrogen bonding that you need in your DNA, in your proteins, creating your shape of your proteins, how your proteins interact with other molecules gets interfered with because you have too much of your H pluses. This is why pH is absolutely key in biology. Starting to make a little more sense why this is important. Okay. But not enough is said about this on the biological side whenever you take chemistry. It's just focus on like the freaking equations, and I'm less interested in that. Yes. So it is important to understand the equation of pH just because it puts into perspective what the hydrogens are doing there and how that affects everything else in the body. So pH is the negative log of H plus. What does that mean? This means concentration when you have these brackets, right? So the concentration of your hydrogen ions, which is your pluses, right? The concentrations of your pluses, your protons, basically in all of this, because when they aren't having their electrons with them anymore, they're basically just protons essentially floating around. I mean, that's a watered down version of it, but these guys are free H pluses. They're not just polar, they're free. They have lost an electron. And so they're now actually H pluses. They're ions that are flowing around. And yes, they will also be H3O pluses when they interact with water, but I'm never going to bring up hydronium ions in my class, okay? So always think of them just as H pluses. So when they're free like that, the concentration of these free guys, the negative log, what that means is negative log means the inverse of 10 to the x. So what it means is like 1 over that. So what we can think of too with this is like if your pH is 2, what this means in the terms of this equation over here is 10 to the negative 2. It's the same, okay? Or pH of 7, 10 to the negative 7. What if I had a pH of 0? It's 10 to the 0, but what does anybody know what 10 to the 0 is? So this is why the pH scale only goes from 0 to 14. It's The 14 is a whole other thing that we don't need to get in an argument about, okay? But it doesn't go below 0 because that's where the 1 is. Like, you can't, just mathematically, that's where just the ratio ends, okay? So it goes from 0 to 14. Don't worry about the 14 part. We can get into that discussion if you really want to, but it goes from 0 to 14. If you can remember that it goes from 0 to 14 and that 7 falls in the middle of that, then you can remember that 7 is neutral. And we can remember too that the lower pH, this is the lowest one, right? Which number is bigger? 10 to the 0 or 10 to the -2? So 10 to the 0 is 1. 10 to the -2 is 0.01. Literally, like, that's the math of it. It's just, that tells you how many decimal points to move it, really, is what that number is at the 10 to -2. Don't overcomplicate it, okay? So if your pH is 2, that means you are 0.01 is the concentration of your hydrogen ions. If your pH is 0, that means that 1 is that means pure hydrogen ions. Okay? So 0.01 is a lower number than 1, right? So if you're at pH of 0, you actually have a higher concentration of hydrogen ions, right? So this is really what pH is measuring. pH is asking you about the concentration of hydrogen ions in a solution. I don't ever care about the bases. I'm never going to go on that side of it, and I don't want to get too complicated with it. But for our purposes, if we're talking about pH, the lower number of pH, yes, it means more acid, but what it really means is more hydrogen ions. So lower pH means more hydrogen ions, and that's what acid really means. So when we have an acid, we have more hydrogen ions. If you have a base, you have less hydrogen ions. And so yeah, pH of 14. Oh my God, I'm not going to write it out. But if you have a pH of 14, it means 10 to the -14. Man, if you looked at that pH of 2, this is the bigger number, right? Literally, like numbers wise. And this is the lower number by far. And if we were to write out 10 to the -14, it would be 0.000000. That's the concentration of the hydrogen ions. And so now we start to see, like, what this really means is really just talking about the concentration of hydrogen ions. And we're just attributing that compared to 7, which is our neutral, which is what plain water has. Plain water has 7. So this is where we have an equal number of hydrogen ions and OH ions. That's what really is going on in water, and that's what pH 7 really is. Equal numbers of hydrogen ions, equal numbers of OH negatives. And if you're shifting that balance is when you get into acid or base. That making sense? We're getting that? Probably if you're sitting here, you probably already get that idea. But I mean, I feel like putting it in that perspective of what the H's are doing at all times, then you can relate it back to why those polarities are so important in biology and how that gets shifted and changed so easily, and why things like vinegar work so well for cleaning things, because vinegar is an acid, and so it's acetic acid, right? So it's a lower pH, it's like pH of like 3.5 or something like that. And so if you have a pH of 3.5, but those cells are used to existing at like a pH of 7.5 or whatever, the bacteria or whatever, and you're lowering that pH to a point where their proteins can no longer fold the way they're supposed to. Their DNA can no longer be double stranded like it's supposed to. You're affecting that pH to the point the cells can't function the way that they're supposed to and interact with water like they're supposed to. They die. And that's why acid is an effective way at controlling microbes. So, but this is just the basics of what I want you guys to understand, because when we start talking about that stuff, it just makes way more sense if you can get this down first. And then we'll talk about the other structures of the molecules as we go through everything. The main biological macromolecules that we have the carbohydrates. It's just a lot like the macros that you eat in your food, right? In your food, you have your fats, you have your carbs, and you have your protein. Those are like your macros, okay? One thing we don't talk about in food is your nucleic acids, because people don't really think about that in their food and their macros. And your body can generally make the nucleic acids for you from those other components, so it's not as important. But in biology, that's your 4th macromolecule, is your like your DNA and your RNA. And it's a pretty easy one to remember if you can remember that all cells have something genetic to them. And so you need your genetics. What's your genetics made out of? DNA. And so remember your nucleic acids. ATP, our energy molecule, also falls into the nucleic acid category. It is adenosine triphosphate. It is literally a nucleotide with 2 extra phosphates on it. We already talked about how phosphates are so negatively charged because those oxygens that are hogging electrons. So when you have 3 negative covalently bonded to one another, they don't want to be anywhere freaking near each other. And we're forcing them to be covalently bonded together. So when you cut off one of those phosphates, it releases energy basically for the cell to use for energy. ATP, easily the most important energy resource in the whole cell. There's other ones too, but they work very similarly to that, GMP and all those things. So, but ATP, the biggest, most important energy resource in the cell, like we said, so important that if I cut off your oxygen, the reason you die is because you run out of ATP. That's why you die. So that exchange of that. And if you haven't taken physiology, one thing that always blows people's minds is when I tell them the oxygen that you breathe in and the carbon dioxide that you breathe out, the oxygen that's in that carbon dioxide never touched the oxygen that you breathed in ever at any point. Not the same oxygen. Most people don't realize that. It came from your food. The carbon dioxide that you exhale came from the food that you ate. It's wild to me that people don't make that connection. So anyways, we'll talk about Krebs cycle and all of that when we get into aerobic respiration in unit 2. But like I tell you guys, I can't stress enough how important chemistry is for understanding a lot of this biology in general, which is extremely essential to just understanding your patients and how their bodies function as well. Not just the microbes, but just overall. All right. Protein structure levels, another one that a lot of people have a lot of trouble with. I wish I could just like make a whole other slide, put it up here. Primary protein level. Structure level is just going to be, this is 1 degree, by the way. I'm trying to write, my handwriting is terrible, I just can't tell. Zoom in a little bit. Primary is going to be the amino acid sequence. Secondary is going to be how the amino acids immediately next to each other. So neighboring. And right next to, we already understand that it's next to it. I did click it, right? What is it doing? It says it's on eraser, but it's not actually erasing where I'm at. It's like way over on the side. You see that? I'm crazy. I'm not imagining this. Yes. Anyways, eraser's not cooperating with me. Neighboring amino acids are groups. Interact with each other. And then this leads to the development of alpha helices and beta pleated sheets. Alpha helices and beta sheets. I'll almost always in the question, I'll almost always refer to the alpha helices and beta sheets. Okay. Almost always, if I'm talking about the secondary structure. Okay. First primary, think amino acid sequence. It's literally like alanine, phenylalanine, guanine, glutamate, that valine, you know, whatever it is. So isoleucine, whatever your amino acid is, that sequence of them is your primary structure. How the ones immediately next to each other interact, literally the ones immediately next to the next guy, can cause the development of alpha helices and beta sheets. Tertiary is where we have the guys that are across the polypeptide, the whole protein, cause interactions. Think of it this way. When you're weaving your ribbon, that's your primary sequence, okay? How the ribbon was weaved can cause it to have little bends in it as you're weaving it. That's the secondary. But then when you tie the bow, that's the tertiary, okay? So one thing you think of it is like if I have a whole bunch of amino acids that are hydrophobic and then a whole bunch that are hydrophilic, polar guys, right? They have charges. So the guys that are non-polar hydrocarbon, I don't know, that sort of benzene ring type of stuff, hydrocarbon stuff, whole bunch of them that don't like water, a whole bunch of them that do like water, a whole bunch of them that don't like water. The ones that don't like water are going to want to fold back and interact with the don't like water guys and expose the water liking guys on the outside, right? Put the water liking guys on the outside. We're hiding out on the inside. We don't want to be anywhere near the water. So that's the tertiary. This is also where we get like cysteine double bonded sulfur bonds, disulfide bonds that are across like the thing. They'll actually covalently bond across from one another in that tertiary structure. And then when you take two bows that you've made and put them together to make a functional pretty bow on your package, that's the quaternary. Quaternary is where you have multiple chains that come together, leading to a functional. Always, that's what it is. Does that help in a little bit? Okay. Questions about the chemistry stuff? All right, because we're going on to bacteria and archaea, I guess. We don't really talk about archaea much. All right, the basics of bacteria, structures, shapes, and arrangements. We learned about the shapes and the arrangements. Really, the only ones that I ever asked about with the shapes are bacilli or cocci. I know there's other ones that the book talks about, but I mean, cool. You guys don't need to know about them for like nursing and stuff like that, okay? So know the cocci and know the bacilli, please. I mean, there's comma-shaped ones and all sorts of stuff. Like Vibrio, that means comma-shaped. And one of the most important pathogens that, well, one of the very important pathogens that we will learn about is cholera. It's Vibrio cholerae. And so it is a comma-shaped one, but like, cool. Okay, so cool story about it. I don't know. Arrangements, really the only ones that you'll have to know of, we kind of already talked about them today, but you definitely need to know the difference between Staphylo and Strepto. A lot of people have problems with this. I understand. They're similar kind of words, okay? So Staphylo, this is where we have irregular bunching. Strepto is strips or chains. That's literally how I remember it, like not even joking. Strepto sounds like strip. And so I think of strip as being like chains. And so literally like when you think of Streptococcus pyogenes, we have strep throat. It's Streptococcus pyogenes. Streptococcus, well, what shape are those guys? They're round, right? So Streptococcus, they literally do this. So I heard my voice like echoing. You're fine. You're fine. You're fine. I was like, that's me. So that's what it will look like. They'll chain up like that in like long chains. Okay. So that's the strepto. Then Staphylo is like in bunches where they're like, we call them grape-like clusters. Okay. Yeah. If you need to go. I'm recording it. So no worries. Capsules, again, they are like sticky coatings. We kind of talked about that for adherence and immune evasion. They're usually sugar-based coatings. Then we have flagella. These are for motility, and they are the only structure that bacteria use for motility. That's flagella. There's different flagellar arrangements that are based on basically the position or how many flagella you have. There's 4 different arrangements. You can have no flagella. Not all bacteria are motile. Staphylococcus aureus, for example, is non-motile. So the flagella can be arranged as mono. It's one word, but I'm going to put hyphens in them. Okay. So monotrichous. Trichous, trick is like hair. That's what that Latin word of that means. So that's really what the trichous part is about. Like if you've heard of like trichotillomania, it's when they pull their hair out. Okay. So trick means hair. So these flagella are hair-like appendages that they have for motility. And what they have, these long hair-like appendages, they rotate to help the bacteria move. Always rotate. They don't move in a whip-like fashion, not for bacteria. Okay, so they rotate. They have a little motor at the base that's protein-based, and the motor rotates it like this. And it either rotates one way or it rotates the other way. And when it shifts how it rotates from clockwise to counterclockwise, it changes its direction. Call it a tumble when it does that. So running is when we're going to rotate in one direction. And then when we stop that rotation and shift it in a different direction, it causes a tumble. And they basically use these runs and tumbles to very, I want to say primitively control how they're moving in directions. And so that's how the movement works for bacteria. So monotrichous means they have one. What the F? Okay, monotrichous means they have one. Lopho means they have a tuft at one end typically just one end. We're going to focus on that. Okay. Amphi means you have flagella at each end. Typically, it's just one flagella at each end. So your cell would look like, if you were amphi, you would look like this. So here's your cell, and then those little squirrely guys coming off are flagella. Lopho, those guys look like this. Got tuft at one end. Mono, those guys just look like that. And then we have peritrichus. This is all over. Salmonella is very well known for this peritrichus. So salmonella looks like this. It has it like just kind of all over, and they kind of like fall like hair behind it as it moves and whatever. E. coli, for example, is a lophotrichus. You don't need to know those. I'm just saying that they're well known for those characteristics. All right. The cell envelope, probably the most important thing that we can learn from this chapter. Not that you don't need to know everything in the chapter, but this is key, man. And we're going to talk about gram-positive versus gram-negative all the time. Okay. So gram-positive, they have a thick peptidoglycan wall. So what the F is peptidoglycan? Well, we have some sugars. I'm going to draw, this is a basic sugar, okay, that are linked together. I'm really bad at drawing too, because I'm impatient. You guys have seen some of the drawings I did. They'd probably impress you, but they're also like not detailed. So there's a reason for that, okay? So NAG and NAM are these, and you don't need to know what they are, what they mean. And I'm never going to ask you about NAG or NAM. You're welcome. And they alternate. NAG and NAM are sugars that'll alternate. And then we have more. If I was doing this in GoodNotes, I could literally just copy this over. I wish I was doing that way, but it is what it is. So same thing. This is the exact same thing. Okay, NAG and NAM over and over and over again. Get our little hexagon sugars here. If a hexagon doesn't look like a shape of a sugar to you at this point, if you haven't, you know, like the chemistry kind of drills that in. I'm never going to have you do like, what is the one that they call like this all in a line? There's like different names for the different structures. I don't give a damn. Okay. I'm never going to talk about that again. Congratulations. So, so I'm going to draw them like this. These are my sugars, and they're linked to each other in these chains. And then these chains are kind of like cross-linked across to one another with these little bridges. And these little bridges, I could have probably done them in a different color. And if you're going to draw this and do it in a different color, but these bridges are peptides. So let me break it down for you. Peptidoglycan. Peptido, the peptide portion. Glycan, the sugar. You guys know that a glyco is sugar. You know this. So these are the sugar portions. I put sugar, but that's all right. Sugar. Put sugar. And this is the peptide sources. Peptidoglycan. So what's a peptide? It's just a chain of amino acid. Okay. So that's what it is. And you see how like this is already starting to build a scaffolding of like sugar chain linked by a bridge. And then that would link to another sugar chain down below it and so on and so forth. All right. So this creates a big scaffolding of sugar chains linked by peptide bridges. All right. And then how thick that is, like how many layers it is, might be a thick wall, but if you have just like one layer, that's the thin wall, okay? And this really what defines the difference between the gram positive and the gram negative. Gram positives have a thick wall. And I mean, we're talking like 30 times thicker than the gram negatives. Very, very thick, sturdy wall. The gram negatives, so if you're a gram positive, you'll have a membrane still containing all whatever your junk is, your DNA or whatever in your cell. So if you're a gram positive versus a gram negative, they both have membranes on the inside. We call that the cytoplasmic or inner membrane. And then the outside, the gram positives have this thick wall all around them. The gram negatives have one thin wall, and then they have another outer membrane around that. Do them in different colors if you need to. So gram positives have a thick wall around their inner membrane. That's it. Gram negatives have a thin wall. Why is that happening? The gram negatives have a thin wall with another membrane on the outside. That helps with like the stability and them being able to survive in environments and things like that. But they're a little bit mushier because of that, right? A lot of our antibiotics affect, not a lot, there's a good group of antibiotics that affect the cell wall. So let's talk about that. Penicillins, we'll just say that, okay? Penicillin, amoxicillin, ampicillin, if it's got psyllin on it, it affects the cell wall. So these are actually going to affect gram positives worse because all they have is a cell wall. They don't have another membrane on the outside to help out with anything. The gram negatives, they're like, I'm barely using that thing anyway. Have at. Take away my cell wall. So psyllins, we typically don't use them to treat. Have you guys noticed that? Like if you have an upper respiratory infection or a skin infection, psyllins. But if you have diarrhea, when are we prescribing a psyllin to a diarrhea? Never. Because gram positives will be like what's in your nose and on your skin. Gram negatives are what's in your gut. And we typically don't treat gram negatives with psyllins because they don't care about losing their cell wall. They'll be all right. It's really interesting stuff when we get into that stuff. It's a chapter 12 thing, but putting it into perspective, why this is actually important in practice, not just for identifying the differences. They have different characteristics because of these walls. So generally speaking, organisms are either categorized as gram positive or gram negative for bacteria only, gram positive or gram negative. You either have a thick cell wall or a thin one with an outer membrane, period. The way the gram stain works is crystal violet is the first stain that you use in a gram stain. You guys will know this like at the back of your hand later on, but crystal violet gets trapped in that scaffolding. And so when you have a very thin scaffolding, you only have one layer or two layers. Crystal violet doesn't really get trapped in there. When we use iodine is the mordant and it fixes it into place. It's really all the iodine does. It fixes whatever crystal violet you trapped in there, it gets fixed in place by iodine. It's mordant. Like think of mortar for like brick and mortar. Okay. So crystal violet gets stuck in the scaffolding in the gram positives. It doesn't get stuck in the gram negatives because their wall is too thin. And then the iodine fixes it in place in the gram positives. There's nothing to fix for the gram negatives. So when we rinse it with ethanol after that, the ethanol rinses out any unbound, unfixed crystal violet. That ethyl alcohol rinses out all that crystal violet from the gram negatives. It doesn't hold on to it. But the gram positives had this big scaffolding network of crystal violet that got locked in place. So you rinse with ethanol and you counterstain with safranin. So suddenly that crystal violet, which is violet, is purple. You've got purple gram positives and red gram negatives. Safranin is red. Okay. And it's an easy way for me to remember too, because gram negative, to me, red is a negative color. In the microscope, it looks pink. Okay. But I'm not going to ever ask you a question. Is it pink or is it red? Okay. I'm going to put red. That's what I'm going to put. Because to me, that's easier to remember. And I hope it's easier for you guys too. So, gram stain, it's either purple or it's red. Red is a negative color to me. Gram negatives are red. So they're going to stain with safranin. They're not, and that already tells you too, if you can at least remember gram negative, that's the negative color, that's the red. Then you can remember, okay, it stained red because the crystal violet didn't stay in it. Why didn't the crystal violet stay in it? Because it's got a thin wall. Remember that. That'll help you remember what the gram stain is doing. You got to know the gram stain for chapter 4. I will ask you lecture questions about it. You also have to know it for the lab, obviously. We're going to do it in the lab on Monday. So hopefully that'll help solidify our understanding with that. On Monday, we'll go into the same detail on it. Okay. The next thing on here is the mycolic acid. Oh, something else that I want to touch on with gram negatives also. So gram positives, because they've got this big scaffolding on them, they have other molecules that help like maintain the scaffolding, and that's called teichoic acids. There's lipoteichoic and teichoic. It's spelled like this, Teichoic. It's either teichoic or lipoteichoic, but they've got teichoic. Okay, and I think of tectonic plates. Tectonic plates to me are big, and so it's the big cell wall has the teichoic acid. All right. Gram negatives, though, on their outer membrane, which gram positives don't even have an outer membrane, right? Their outer membrane has something called lipopolysaccharide, or LPS, also known as endotoxin. Extremely important for the future that we know this, because we're going to talk about endotoxin a lot. And one of my favorite things to do is ask you guys about like, hey, your patient is sick. They have this blah, 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 blah. And this tested blah, 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 blah. And they had a test that showed that they have endotoxin. Which, of these could it be? Could it be Staphylococcus aureus, which is gram positive? Could it be this, which is a gram negative? Could it be this, which is not a bacteria? Da, 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 da. But you'd have to know just by the fact that it has endotoxin. This is something on gram negative outer membranes. All gram negatives have this on their outer membranes. This is what leads to shock associated with blood infections caused by gram negatives. So your body is naturally inclined to respond to this with an inflammatory response. Regardless, it's non-specific. If it sees endotoxin, it doesn't care what organism it is. If it sees endotoxin, that doesn't belong in your bloodstream, and you have an inflammatory response to it. So this is a huge trigger in healthcare stuff. So it's really important to remember endotoxin. If you see endotoxin, think gram negative. If you think gram negative, think, oh, they have the outer membrane, thin wall, endotoxin. And I'll always remind you in the lectures, I'll be like, endotoxin and LPS. Endotoxin and LPS are the same thing. Okay. All right, now we'll move on to mycolic acid. This is a wax that is in the gram-positive wall in only certain bacteria. Okay, so the only bacteria that can do this, there's only two genera. They're either called nocardia or mycobacterium. And I will never ask about nocardia. It's in the textbook, okay? But I won't ever ask about it because to me, I want to know about the medically relevant guys. And mycobacterium is medically relevant. Mycolic, mycobacterium. There's a problem that we're going to get to here in a second with the bacteria and the myco thing in a second. But for now, if its name is mycobacterium, focus on the myco on this one. It's a bacteria. Yeah, so what? A whole lot of them are. This one has myco, okay? This is mycolic acid. It's a special wax. It makes them hardy and resistant to things. And it's on the surface of the wall. And it's really hard to stain it. It's resistant to stains and all sorts of stuff like that. And we have a special acid-fast stain that we do that detects mycolic acid. We use carbofusion, which uses carbolic acid, which is phenol, that is like going to be able to bind with those hydrophobic waxes. Waxes are lipids, so they're hydrophobic. So the stain binds with those waxes and then doesn't bind with other cells that don't have that wax. And so if you're a mycobacterium, you'll stain with carbofusion, that stain. But if you're not a mycobacterium and you don't have the wax, you won't stain with it, period. Like that's just how it works. So only bacteria that are called mycobacterium, blah, blah, blah. Not Staphylococcus aureus, not Escherichia coli, not Bacillus anthracis, none of those. Only if it's named Mycobacterium, blah, blah, blah. For example, Mycobacterium tuberculosis, Mycobacterium leprae, okay? Tuberculosis and leprosy. Leprosy is extremely slow growing. It takes a month just to get one cell to double itself, which is wild. When we're talking about E. coli, it'll double itself in like 20 minutes. But a month for leprosy to double itself is extremely slow growing. Tuberculosis is also kind of difficult to grow. It's also a BSL-3 pathogen, so it requires special, containment procedures and stuff like that. But I want to talk really quickly about tuberculosis, which I feel like is underappreciated in the world. 1/3 of the world's population is infected with tuberculosis right now. 1/3 of the world's population is infected with tuberculosis right now. So it doesn't need to be appreciated. Do we have a vaccine for this? Yes. That should put into perspective how contagious it really is. Huge, huge issue with antibiotic resistance. It's really antibiotic resistant, but let me tell you why first, because we know that probably the biggest issue with antibiotic resistance is patients not finishing their antibiotics the way they're supposed to. It's why you need to finish your prescription when you're prescribed. Because if you don't kill off everything. Any of them survive. They could come back more resistant, and then you're screwed the next time we use that antibiotic. It's why we have gonorrhea that can't be treated anymore. So that's a problem, right? Same thing with tuberculosis. It can still be treated, but we are running out of options because tuberculosis lives inside of your cells. It's one of the bacteria. Most bacteria live like outside of your cells, but Mycobacterium tuberculosis actually lives inside of your cells. And guess what cells it lives inside of? The immune cells that attack them. So it gets eaten up and it's like, thanks for the house, guys. This is great. And it lives there. And then your body tries to wall it off and it creates these nodules in your lungs where it just like hides out and can live there for years and years and years without any signs or symptoms. But it comes back out and people start coughing up blood. That's consumption. And so that's what we call secondary tuberculosis. And so people are often infected with tuberculosis and don't even know it. We've had several outbreaks recently here in Oklahoma, if you haven't been paying attention. Even in like Edmond public schools and stuff like that. There's an outbreak in Kansas recently in public schools. This is why they test you when you go work in a hospital, because you just don't know sometimes if you have it. But it's 6 to 9 months of antibiotics. How many of you guys stick into that? People can't even stick to their freaking 10 days. So, and then a lot of people that have this, they also have AIDS or they'll be homeless or something like that. How many of those people do you think are sticking to 6 to 9 months of antibiotics? Yeah. And these are antibiotics that are hard on your body too. So no wonder that people aren't sticking to it correctly, especially in other countries. This is a huge deal in Russia. And so whatever. So I feel like I didn't want to not talk about that. And we'll talk about tuberculosis more and more throughout the semester, but you can't leave it out. All right. So that's what mycolic acid tests for. So one of the biggest things you can do is the acid fast stain. If you have a patient that's coughing, you'll see this all the time, especially if you do a rotation through the ER at Integris Baptist. We got it all the time. You get homeless people that come in with a cough, you get their sputum, send it up to the lab for an AFB, which is an acid fast bacilli stain. And we'll just do a stain on the sputum straight up and look for acid fast bacilli because only mycobacteria are going to have that. So that's tuberculosis. If you're acid fast positive, you have tuberculosis, period. So there's that. Easy, quick. You don't even have to culture it. So cool stuff. All right. The next one, the ones that don't have a cell wall. We have some bacteria that they don't have a cell wall at all. Like they might be like the gram negatives, but no wall at all in the middle. Okay. So they might have two membranes, but no wall. There's only one genera of bacteria that has this. And it unfortunately is also a myco. So this one is called mycoplasma. Mycoplasma. So the way I remember this guy, if I've got a myco something bacteria that my professor's asking me about, there's mycobacterium or there's mycoplasma. So I'm like, okay, it's a myco. Is it plasma or bacteria? If it's bacteria, then what I care about is the myco. If it's plasma, then what I care about is the plasma. And so this is how I remember this. If you guys know about plasma itself, right? If I take your blood and I separate it, the plasma is the liquid portion, right? So if I'm carrying around a cup of plasma and I spill it on the table, will it hold the shape that it was in the cup? No, it's just like water, right? It goes everywhere. Same thing with these bacteria. They don't hold their shape. So they are pleomorphic, many shape. And that's because they don't have a cell wall. Just like the cup needs to have the shape, hold the shape for the plasma, the wall needs to hold the shape for the bacteria. Okay, so that's literally how it's stupid, but that's how I remember this one. All right, so mycoplasma, I'm like, oh, that's the cup of plasma one. The cup held the shape. The wall isn't holding the shape. That's what's going on here. Okay, so that's how I remember this one. So if you are a mycoplasma bacteria, so these are medically relevant as well, which is why I'm even talking about it, but Mycoplasma pneumoniae is the cause of walking pneumonia. So it's not the number one cause of pneumonia. That's Streptococcus pneumoniae. Okay, but the number one cause of walking pneumonia, which is atypical pneumonia, it's like you have pneumonia, but you're still able to get up and ambulate and things like that. That's walking pneumonia. That's caused by Mycoplasma pneumoniae, hands down. And so it's a mycoplasma. This is a defining characteristic of these bacteria. All right, so we already talked about the gram stain, right? We talked a little bit about endospores. There is a better picture than what I'll ever be able to draw on here in like the notes in the textbook for this, like the cycle between endospore and vegetative. But it is important that we know those things, right? So with the endospores, the vegetative is the active, dividing, reproducing, whatever you want to call it. Metabolically active, why? Whatever, if it's doing stuff, it's vegetative. If it is endospore form, it is dormant and inactive and inert. But what's its point when it's doing that? Just to be hardy, just to wait things out. Okay, so it's hardy and resistant, extremely resistant. Can't survive an autoclave, but it can survive pretty much any other cleaning method that we have. Can't survive incineration, but it'll survive boiling. Okay, can't boil an endospore away. Boiling, by the way, doesn't sterilize. So you can't boil out an endospore. Not all bacteria can make endospores. There's only 2 genera for this one. So these are some flashcard shit right here, right? So we've got mycobacterium that are our mycolic acid. We've got mycoplasma. Those are the no cell wall. And now we've got our endospore forming guys. These are only going to be Clostridium and Bacillus. And I'll try to keep it that straightforward for you for the course. There are some that are like lactobacillus or something like that, or like one that we're doing in the lab is Lyscinobacillus. But they've got bacillus in the name. It's almost certainly going to be making endospores. Like when you take like your probiotic supplements that have the bacteria, if you've ever like read the back of them, how many of those have bacillus somewhere in the name? Because they're endospores that can survive your stomach acid. So it might be lactobacillus, but it's a bacillus. So bacillus and Clostridium, these are the only ones that we worry about that can make endospores. No other bacteria can make them. So it's not like any of them can make them if it's harsh enough. No, only Clostridium and Bacillus can make endospores, but they're pretty widespread. So that's a problem. But yes, all cells, if they're active and dividing, will be considered vegetative. Only Bacillus and Clostridium can turn into endospores though. So the process of going from a vegetative happy dividing cell into an endospore is called sporulation. The process of going from the spore, endospore, and turning back into a vegetative cell is called germination. Those terms are in the text and I could go over them more, but I mean, they're pretty straightforward. Just take the time to know what they're called, steps, okay? And then we can sort our eukaryotes based on cell envelopes. There are 4 groups of the eukaryotes, all eukaryotes, is bacteria and archaea, okay? So this will be firmicutes, gracilicutes, there are tanaricutes, and there are mendozicutes. All right, this is how I break it down. Are we ready? Firmicutes, they're firm. Which one do you think would be the firmest of the ones that we've talked about? I would hope you would say gram-positive. They've got those thick walls, right? So these are gram-positives because they have the thick wall. It's all based on their cell envelopes. So whatever's on their outside, holding them together, okay? So our next ones are gracilicutes. I remember this one because of the G. If our gram-positive is the firm one, what do you think the gracilicutes are going to be? Gram-negative. Just that's how I remember it. All right, tanaricutes. Tenar, Latin, basically for tender. Also, you can say that tenar looks like tender. I say that. Let's say they're tender. What are the most tender? If it's not the gram negatives, well, these are the ones without the cell wall. Literally, the only ones that we talked about were the mycoplasmas. And then the mendozicutes. This is where it gets a little weird, okay, guys? When I was in middle school, I had a boyfriend named Joey Mendoza. That was a long time ago, guys. I would say that was even archaic. All right, so these are the archaea. Joey Mendoza. That's how I remember it, and that's weird. And if it helps you guys remember, man, Professor Yost had a boyfriend in middle school, and his name was Joey Mendoza. That was a long time ago. That's how I remember that the mendozicutes are the archaea. It's weird, but it helps me remember it. If there's a better way you guys have of remembering those things, please use it. But like that's how I remember it and divide it out for you guys. I wasn't born a master of remembering this crap, so that's how I learned it. Okay. Do you guys want me to go over the questions, or is it going too long for you guys? Do you have time for that? Any of that? Because I can leave it for you guys. The questions aren't like, no. We've got germ theory, just what it is, the fact that it was developed by Louis Pasteur, and you will be asked to identify the old guys on the exam. I feel like that's pretty straightforward. You guys get used to expecting that. I don't care about it either, but I have to test you on it, okay? So remember that's Louis Pasteur who developed the and tested germ theory. It was Pasteur. What did Lister do? Lister did the aseptic technique thing, right?
To know about the one, the Tyndall guy.
Oh, Tyndall. Yeah, so we won't learn about him until chapter 11. And I'm not going to ask you about him.
Okay.
Yes, but Tyndallization. So he actually, don't let me get off on that. I'm going to talk forever. Okay. So so Lister did aseptic technique. So you definitely need to know him right now, right? Jenner, he developed the smallpox vaccine. We haven't really gotten into him too much. I think I like briefly mentioned him that one day about vaccines. And then Einstein has nothing to do with microbiology. Plague doctors stuff there. We already talked about this too. Stuff their nose with stuff. Why was this inherently ineffective? This is just referring to the fact that this doesn't block the germs. It just blocks the smells, right? So smells don't transmit illness. Germs do. If the herbs were antibacterial, even if they were antibacterial, does that inhibit like fungi? Does that inhibit? No. So even if they were antibacterial, it might protect you from some bacteria, but it's not. It's ridiculous. So this is like one of those things that, are any of you guys in the nursing program already? Yeah. So this is the type of crap that you're going to see on the nursing exam all the time, right? Which is the best answer? So can antibacterial things sometimes help with bacteria? But don't forget, we're not only talking about bacteria can cause disease. Don't let it trip you up, right? So that's the kind of question you'll see a lot with that. Eukaryotes lack a nucleus. U means true nucleus. So knowing the difference between eukaryotes and eukaryotes, and you guys are welcome to go over these slides and look at them in a little more detail. I'm just kind of pushing through so we can get done. But mitochondria, we think, evolved from like a cell eating up another cell. And there's so much evidence for it. It talks about it in chapter 5, so I'm not going to get into detail in it right now. If you can't have eukaryotic cells without mitochondria, what can you infer? So we had to have cells before we could have eukaryotic cells. And, no membrane cells had to exist and come together first, right? So we had to know that bacteria had to come before the eukaryotes. So not bacteria evolved from eukaryotes. Not this was opposite. Eukaryotes existed before bacteria. No, it's the same as A. Bacteria existed before eukaryotes. Yes. And then eukaryotes evolved from archaea. This is not talking about this, it's talking about bacterial symbiont. You see how that's the hint there. Don't forget it. Looking at a microscope, you see a large cell with several membrane-bound sections. If I'm already talking about specifically a membrane-bound cell, you know you're not talking about a bacteria, and you're not talking about an archaea. It's not one of those. We know for sure. What else can we say? We already know it's a cell. If it's cell-based, it can't be a virus. Uh-oh. So I've already answered the question just by thinking about what's in the question already. What type of cell could this be? Yeast, because it's not archaea, it's not bacteria, and it's not a virus. Especially in chapter 5, we're going to learn that yeast are fungi, which are a type of eukaryote. All right, selective versus differential. Remember that selective selects for. That's really, it's in its name. And differential just shows you the difference. So selective selects for or against, and differential just shows you the difference. Nobody's being favored there. We have plenty of media that do both. So we'll learn about that, like McConkey agar and mannitol salts agar do both. So it's important to remember that as well. So which of the following is an appropriate definition of selective media? So selecting for or against, right? So some things will grow and some things won't. Media that changes colors. No, that's not allowing some things to grow and not others. Media that favors growth of some organisms. Yes, growth of some, inhibits others. This is looking pretty good, but don't be that person, right? Don't be that person that selects an answer and doesn't read the other choices. Don't be that person. So I even do this in my own questions that I already know the answer to when I go over them. Media that selects for organisms, this is still dealing with, would require added nutrients. So this is one that I didn't talk about, I forgot to get into because we talked about selective differential. This would be an enriched media. Enriched media have growth factors. It's anything that the organism needs to live that it can't make for itself. This includes things for us, right? B12, vitamin B12, most of our vitamins, we can't make them ourselves. We have to get them from our diet. And so if you can't make it for yourself and you need it provided, that's a growth factor. And you already know we have foods in human diet that we call enriched. You can buy enriched, your cereal says enriched, your bread says enriched, whatever. It has, God forbid, it has riboflavin in it or something like that. It's vitamin B6, just to be clear, right? So if you have vitamins that are added into something, those are growth factors added into the media that you consume. That makes that media that you consume, your bread, your cereal, enriched, right? You are an organism that requires growth factors. That term is fastidious. Fastidious applies to the organism. Enriched applies to the media that has the growth factor in it. If you need a growth factor, you are fastidious. Okay? Humans are fastidious. We eat our enriched food. All right. Media that kills all bacteria except for one type. It doesn't have to necessarily kill all bacteria except for one type. Would that be selective if it did that? Maybe, but is that the best definition for it? No. So that's where you guys already know if you're in a nursing program. So yeah, so B is our answer. McConkey agar, it has bile salts that favor gram negative. Ooh, so we've got something in it that is selective. And the media contains an additive that causes bacterial growth to turn pink. Ooh, a color change because it can do something metabolically. That's differential. So this isn't just selective, it is selective and it is differential. So it's both. Okay, easy kind of question, just be able to break them down. Yeah. We already talked about the growth factors, so I'm not going to go over that one. I'm actually going to leave these questions here and see if you guys can answer them because we just went over this stuff. So I'm going to move on to the next topic. Differential stains, the gram stain, the acid-fast stain, the capsule stain. Does the organism have the feature or not? That's what a differential stain is. So it's literally staining for a specific feature and whether or not the bacteria has it. Okay, so which of the following stains would be differential? So we talked about media being differential, but stains can be too, and it's still dependent on colors. So easy enough, right? This color or that color means positive or negative. Staining for the presence of flagella, that would be a differential feature. A stain using one dye that stains the cells one color to create contrast. That's like what we did today. That's a simple stain. It's not differential. It's not telling you a difference between anything. A stain which kills the bacteria first, that's not telling you a difference between anything. Staining a slide with one dye and another slide with this with the same organism, but with a different dye and comparing them. No, it's a one staining procedure that can tell differences from whatever your color is, like the gram stain. I put all of the cells through crystal violet, iodine, alcohol, safranin, and whatever color you are after I do those 4 steps, purple or red, tells me a difference. That's a difference. It's one staining procedure, and you get possibly different results that tell you different things about the organism. So that is A. Mycolic acid is a wax on the surface of acid-fast bacteria. Why might you guess that a special differential stain, carbofuscin, would need to be used? Carbofuscin must be a positive stain. Remember, positive stains are going to be the stain itself positively charged, and the surface of your cell has these negative charges on them, and these attract. Is that happening with a wax, which is a lipid? What do we know about lipids? They are hydrophobic, right? Because they have long carbon-hydrogen chains. Whatever, I could drop forever, right? But you know, they have these long hydrocarbon chains. Everybody's very happy. Do we have hydrogens in them? Yeah. But are those hydrogens positively charged? No. Because why? Because carbon plays fair. It shares the Barbie equally. Oxygen doesn't do that. So when you have these long hydrocarbon chains, everybody getting a fair Barbie, ain't nobody got a charge. And so they are non-polar. They don't play well with polar things. So that's what a wax is. It's a lipid. All right. So if it's positive chain, this stain, this is a charge. Our lipids aren't going to do well with that. Carbofuscin, is it lipid soluble? Easy enough. Carbofuscin must be heat activated. We never talk about anything being heat activated necessarily, but that I don't know why that would help with it. Carbofuscin must be a negative stain. Again, you're still talking about a charged stain. So this is definitely going to be the lipid soluble thing. Waxes are lipids. We learned that in chapter 2. The gram stain, we'll see if we can answer questions with what we talked about with the gram stain. We know, I'll write up our notes at the top just to remind you guys, we just talked about it. Gram positive, we have a thick wall. And we're going to stain, do you guys remember what color? Positive is purple. Because negative, the negative color is red. All right, so we got a thin wall for the gram negatives and an outer membrane. And it is red. Purple positive. All right. So what is the difference in gram positive and gram negative bacteria that makes them stain the way that they do? Is due to different charges on the cells, the presence or absence of LPS. Don't get hung up on this. Do gram negatives have LPS? Yes. Do gram positives have LPS? No. It is a difference between them, but is that what the gram stain is based on? No. Presence or absence of a nucleus, not what we're checking for. We're checking for differences in the bacterial cell envelope. And what the cell envelope means is just whatever's on the outside of it. And that's what that is. So D is our answer here. Patient sample is sent to you testing for the presence of a suspected gram negative bacteria. Okay, so if it's gram negative, what color are we going to expect it to be? Red. We'll write that out to the side first. After performing the stain, the cells are purple. What do you report to the medical team? The patient sample is in fact gram negative. They were correct. The patient sample is gram positive. They were incorrect, probably this one, but let's see what the other choices are. They must have sent the wrong sample, or the gram stain is inconclusive and will need to be repeated. I mean, it says you performed the stain. It doesn't have any indication that we did it incorrectly. So we're going to assume that they were incorrect. I know you thought it was gram positive, guys, but it turns out it's gram negative. Maybe that's weird for this situation. I don't know, but it's this. B. God, but we're only in chapter 2. Let's see if we can do what we can. All right, so let's see if we can answer some questions regarding to the chemistry stuff. If magnesium and sulfate allow it to dissociate into Mg plus and SO4 negative. I'm not balancing these equations clearly, but that's okay. All right. When dissolved in water, what type of bond exists between Mg and SO4? If it's got a plus and it's dissociated from the SO4, which now has a minus, that minus is because it gained an electron. Oh my gosh, I'm not even bringing up redox, but we know what's going on here, right? So something donated an electron to something else. What type of bond is that? Ionic bond. Transfer or donating electrons there. If they're sharing the electrons, they're staying bonded. They're not going to dissociate into a plus and a minus. Covalent bonds share electrons. Ionic bonds, we have an actual transfer of an electron to something else, and they'll actually dissociate because of those charges in water. Van der Waals, don't talk about it in my class. I don't even want to hear about it. And then hydrogen bonds are just attractions. Okay. DNA exists as a double helix, and it's called double because there are 2 strands held together exclusively by hydrogen bonds. In any one of those given bonds, what type of electron interaction is occurring? Do we have electron sharing, electron donation, or just polar attractions, or negative electrons becoming positive? That's it. Electron sharing is covalent, electron donation is ionic. No case ever where negative electrons become positive unless you're doing some weird stuff in like a hadron collider or something. I don't want to know about that either. Don't tell me about physics, okay? All right, now let's move on to our chapter 4 stuff. This was our bacteria, where we're getting just a little more detailed on the gram positive versus gram negative, those chemical structures involved there, as well as the flagellar arrangements, and learning about peptidoglycan. So this is getting more into the actual structural difference between gram positive and gram negative cells here, and talking about the peptidoglycan and that stuff. So patient sample is tested and found to have lipoteichoic acid. Remember how we talk about teichoic and the tectonic plates, and that's a big structure. And what's got the big structures? Gram positives, right? Big thick cell wall. This is a gram positive bacteria. It's not gram negative. They don't need it. They don't have a thick cell wall that needs help being maintained. That's what teichoic acids do. It's not acid fast. It might be. Interesting. So acid fast bacteria that have that wax on the outside, that's only going to be in gram positives. So it could be, but is this telling us whether it's acid fast or not? No, but we do know for sure it has to be gram positive. Which of the following is not a feature of the gram positive outer membrane? Do not let it trick you. This is a trick question. Gram-positive outer membrane. They don't got one. Okay. Gram-positives have lipoteichoic acid in their wall. Lipopolysaccharide. This is something found in the gram-negative outer membrane, but not in the gram-positives. Peptidoglycan found in the gram-positive wall, but they don't have an outer membrane. Okay. Trick question. Yes. All right. Say if you're paying attention. All right. Next one is talking about capsules. These are the sticky sugar outer coatings that are associated with adhering or creating biofilms or with evading our white blood cells from eating them up. That process is called phagocytosis. You're going to hear that word so many times, it's going to make you sick. All right. Example question. A bacterial strain has been genetically modified to do a capsule with altered polysaccharide composition. No, stop. So we got a weird capsule going on here. Researchers observed that the modified bacteria show reduced virulence. What does that mean? Decreased effects of infection, decreased disease. They aren't as able to cause infection compared to the wild type strain. Based on your understanding of capsules specifically, which of the following best explains the observance in the reduced disease causing aspect? Okay. Does the altered capsule composition prevent the bacteria from adhering to the host tissues? Maybe. Modification enhances the ability to evade phagocytosis. I imagine if that organism could evade white blood cells better, it would be causing more disease, right? So it's probably not B. The altered capsule disrupts the bacterial cell wall. This is not how capsules work. Okay. I'm just going to move on. But would that lead to increased susceptibility to environmental stresses? Potentially if you're disrupting the cell wall, but that's not what capsules do. They're just a sugary coating outside. Okay. The modification inhibits the synthesis of exotoxins by the bacteria. I don't know if that's right or wrong, man. I don't have any idea. I don't see how, but what we know about capsules and what they do with adhering and things like that, A is our best answer here. It's the clearest. Now, you see me on these last two choices being like, I don't know, man, this doesn't really make sense. I don't think that this aligns best with our choice, but how can I really say, I'm not gonna have choices like that on the test. These questions are written to be like really getting you guys thinking about how to apply this stuff. Now, will it be a little hard? Yeah, it'll be hard, but they won't be like questions where I'm like really taking it to like, you really are meta thinking like all the equations around your head type of stuff. Okay. Because I feel like those choices were kind of getting a little in there. Next, I write these questions like this on purpose. They're meant to be really challenging like that to make sure you guys are really understanding the material and can apply it. In a laboratory experiment, researchers treat a bacterial culture with a chemical that degrades polysaccharides but leaves proteins intact. So capsules are made of sugar and sometimes have proteins too. Sometimes. All right. After they treat it with this chemical that degrades the sugar, this bacteria can no longer evade phagocytosis, no longer escape the white blood cells. What conclusion can we draw about the role of the capsule in this context? The capsule's polysaccharide is crucial for evading phagocytosis. Probably, we just messed it up and it can't do it anymore. So it's probably this. The protein component might not be involved in the ability to do that. Sure. Protein component of the capsule is essential. No, we didn't affect the protein. Both polysaccharides and proteins are essential. I don't know, but it doesn't seem like we didn't affect the proteins. We didn't even test that. So it doesn't really apply to this. The chemical treatment suggests that the capsule does not play a role in ability. No, it definitely did. We had a change in it. So it's A, the sugar component is important for evading. Okay. Is this the last one? Please tell me it's the last one. There's one more after this, isn't there? I bet there is. Okay. All right, whatever. Endospores. We know about this. This is endospores. Okay, we have a vegetative cell that does everything. Yay, I'm alive and I do things. And we will cycle into this hardy, dormant endospore that just waits it out until things get good again. This process is called sporulation, going from vegetative to endospore. And this process of waking back up again is called germination. I'm just gonna draw that out here real quick so I have something to refer to. Picture is better in your notes. Okay, research discovers a bacterial strain that forms endospores. Do you guys remember which genera? You're gonna try to, like, you're gonna call it out at the top of your head on the freaking like third day of school. It is, by the way, Clostridium or Bacillus. I'm just gonna write those up here too, so we can refer to that. All right. Researcher discovers a bacterial strain that forms endospores. So it's either Clostridium or Bacillus, has to be, but lacks certain genes associated with spore coat synthesis. So that's the thing that makes it hardied out here, because it's like it basically like takes its DNA and condenses it down and then puts this massive coat around it that is like resistant to everything. So if it's got a problem with this coat, it might not be as hardy or resistant as it's supposed to be. All right. After exposure to extreme heat, the strain shows reduced survival. So apparently that coat was important for resisting heat. Which of the following conclusions can be drawn? The bacterial strain's reduced survival is due to a lack of DNA replication. This seems to affect the coat's ability to resist heat. The absence of the spore coat genes impacts the ability of the endospores to withstand extreme environmental conditions like heat. Okay. The reduction in survival rate suggests that bacterial strains endospores have a higher metabolic activity, and they would grow better if they were higher and faster. The strain's endospores are likely to be more resistant to chemical disinfectants. More resistant to chemical disinfectants. I'm not seeing that at all in what evidence we were given here. So it's going to be B. Patient of yours is diagnosed with C. diff. Why would containment protocol be more strict for C. diff than, let's say, for Salmonella? Is it because Salmonella is endospore forming? No. Is it because Clostridium difficile is? Yes. And this thick coat makes it resistant to a lot of the cleaning agents that would normally get rid of things like Salmonella and whatever else, but it can't get rid of C. diff. All gram-negative bacteria are endospore forming? No. Gram-positives can be, and we haven't even touched on that. Both Clostridium and Bacillus are gram-positives. Really doesn't have to do with the fact that they're gram-positive, it just has to do with the fact that Clostridium and Bacillus can make endospores, and they happen to be gram-positives. No gram-negative ever makes an endospore, definitely not all of them. So all diarrhea causing bacteria are endospore forming. Clearly, it even tells us that isn't the case. Okay. In fact, most of our diarrhea causing bacteria are gram-negatives, and they don't make endospores. C. diff is just, taking it next level. What is this even talking about? The cell, okay, this stuff, Mendoza-Cute stuff, right? Okay, let's see if we can remember, right? All right, so we'll write it out to the side on our choices first. Firmicutes, do you guys remember what those were? Gram-positives, firm because of the thick cell wall, right? So that makes the Gracilicutes gram-negative, which are other G1, right? Mendoza-Cutes, these were archaic, like my old boyfriend, Joey Mendoza. And then Tenericutes, tender because they have no wall. These are our mycoplasmas. All right, so our question is, microbiologist is studying a bacteria that has a thick peptidoglycan layer in its cell wall. Based on this description, where does it belong? Firmicutes. Okay. Now I got a real question over here. 56-year-old patient presents to the hospital with severe GI symptoms, including nausea and whatever. Lab tests revealed that the causative agent is gram-positive. Okay, so we got a gram-positive. It's rod-shaped. What's that going to be called? Bacillus. Upon further testing, it is noted that the cell wall is thick. It shows resistant to desiccation in high temperatures, often associated with gram-positives, but also especially with those that make endospores. The physician suspects the infection might be due to a common bacteria in the soil that can form endospores. Okay, so probably Clostridium or Bacillus, capable of surviving extreme conditions. Based on the case details, which of the following bacterial groups is most, well, this is easy enough. It's Firmicutes. All right, we good? We made it. It went over time. I figured it would on our first one because I'd like to go in a little more detail on that review. Any questions? Good. Okay. If you think of any, always email me. We've got the weekend. Remember to get your stuff done. I will send out an email reminder tomorrow morning. You'll get an automated message reminding you what needs to be done by Sunday at midnight, as well as what needs to be done before our next lab review. Next lab review will be on Tuesday. I'll just be doing chapter 5 and 7. So the eukaryotes and the viruses. we'll do a review right after lab on Tuesday, join or not, I'll record it either way. And then Tuesday night at 8 p.m., we'll be doing the Kahoot game review of everything. And then on Wednesday, we'll have the exam. Yep. Okay. See you guys next time.
There's not gonna be any select all that apply, right? Is that a form of question?
There are some that are like that. I mean, they're not like majority like that. There might be a few questions that it'll be like, but you mean like you have to actually click like multiples? No, I don't do it like that. It'll be one answer is correct.
Oh, okay.
One click. So you won't have to do, don't say that to like future semesters because I might change that, especially for the lab, because the lab has a lot of stuff where I'll be like,
Versus
that. For you guys, it'll be one. For you guys, it'll be one for my class. Yeah. But one of the answers might be all of the above, but you know what I mean? Yes, maybe. Yes. But like I said, that won't be a majority of your questions. Most of them will be, I feel like, straightforward. Yeah.